Sorry. Yeah.
Which unit?
This is last topic. Fourth one, organogenesis. Don't you have syllabus copy?
Yes, ma'am.
So what's the problem I linked with you?
Based on my textbook.
I don't have the textbook right now. See, when I'm explaining the slides, how can I see the textbook? So you, if you have, you just listen. First listen the concept, then you can go to the textbook and read. Okay?
Okay, ma'am. Thank you, ma'am.
Yeah, yeah.
Okay, ma'am. Thank you.
Yeah, yeah.
Sorry for
Okay.
disturbing.
It's okay. It's okay. So, when these acrosomes will go and digest the zona pellucida, okay, because they have digestive enzymes, they will penetrate through the zona pellucida and then they will release its genetic material into the egg. Then the egg will start activating. Until that time, egg will be in inactive state. So when the genetic material is released into the egg, then it will start activated and then the fusion of nuclei takes place and then the division starts. So here, so the sperm penetration and membrane fusion. So here the protective layer of the egg includes zona pellucida, vitamin envelope. So this vitamin envelope is seen in sea urchins, okay? Whereas zona pellucida is seen in mammals. This is the protective layer of the egg. So why they are protective? We'll see. And the acrosome, which is present in the sperm, that contains a digestive enzyme, which will enable the sperm to tunnel its way into the egg cell membrane. Now the membrane fusion permits the sperm nuclei to enter into the egg cytoplasm. Now then the egg will become activated. So till that time, egg will be like usually in females, the number of eggs which are required for the entire lifetime, they're already present from birth, okay, from the time of birth. So usually they all will be in inactive state. In the in the metaphase of the second meiotic division. So when this sperm releases its nuclei, the membrane fusion triggers the egg activation by releasing the calcium. Okay, therefore, this results in the metabolic and physical changes in the egg. So then, this, you see, we all know that many million sperms will be competing to fertilize the egg, but only one sperm can fertilize an egg at a time. Okay, so to stop the polyspermy, why it has to stop? Because if multiple sperms will fertilize the egg, then what happens? The genetic information or genetic material will be in many copies. Okay, so that zygote won't survive. It won't grow. Okay, it will get aborted. So only one sperm has to fertilize the egg at a time. So to prevent this polyspermy, the membrane potential of the egg will change and the exterior coat of the egg will change and the enzymes from cortical granules will remove the sperm receptors which are present in the zona pellucida. So here we can see this is the structure how it looks like. So at the center this is the egg. Okay, at the center we have the nucleus of the egg and which is surrounded by the cytoplasm and towards the exterior surface there are cortical granules, small round granules, and surrounding that we have vitelline envelope, then plasma membrane, then jelly layer, zona pellucida. Okay. Now, whenever the sperm enters this zona pellucida, only one sperm can fertilize. Then, once one sperm fertilizes the egg, then the zona pellucida will become harder so that it will prevent the entry of another sperms. So, This is the original image, how it looks like. Okay? Now, after activation, this egg will trigger it to complete its meiosis, complete meiosis, and the cytoplasmic contents will rearrange themselves, and the protein synthesis and metabolic activity will sharply increase. So this is how in the diploid nucleus, the primary oocyte usually will have the diploid nucleus. Oocyte is nothing but the egg. Okay. Then it enters the cell division, first metaphase, then it will undergo second metaphase, and it will get blocked in the second metaphase state, metaphase of the second meiotic division. And we'll get the pronucleus, which is haploid. Now this pronucleus will be participating in the fertilization. The haploid sperm and haploid egg nuclei now migrate towards each other, and they will form a microtubule based aster, and they will fuse. Now both are haploid, and after fusion they will become diploid nuclei, and that cell is called single cell with the diploid nuclei is called as zygote. So this is how the entire fertilization looks like for your understanding. Like sperm first penetrates through the granules of cells, and it will penetrate into the zona pellucida, where the acrosomal enzymes, you can see small dots here, okay, white dots, they are enzymes. They will digest the membrane of zona pellucida, and they will release their genetic material into the cytoplasm. Once the sperm releases its genetic material into the cytoplasm, the other part of the zona pellucida is thickened by the cortical granules release enzymes. Okay, that enzymes will harden the zona pellucida, and all the receptors which are present for the sperm entry, they will be removed. Now this nuclei will fuse with the X nuclei, and it will form a zygote with a diploid nucleus. Okay, this is how the fertilization takes. Once the zygote is formed, then the next step is cleavage. Cleavage is the rapid division of the zygote into the larger and larger number of the smaller and smaller cells, which are called blastomeres. Here the thing is, all the cells are divided within the same membrane. Okay? There is no increase in the size of the embryo, but within the same embryo, the number of cells are increasing. And the cells are called blastomeres. And once the embryo is formed, the embryo forms two ends. One is animal pole and other is vegetal pole. Animal pole forms external tissues, and the vegetal pole forms the internal tissues. So the outermost blastomeres, they are tightly linked to each other, tightly bound to each other by tight junction. And innermost blastomeres, they pump sodium ions into the internal space, and they will create some osmotic gradient. Okay. So now, finally, the ball of cells which are formed are called blastula stage, and it will have, whenever the sodium ions are drawn inside, it will fill some water. It will pull some water from the outside, and that water forms a fluid-filled cavity which is called blastocele. Okay? So this is how it looks. In sea urchin, you can see the amount of yolk is very little. So in the last class, we have discussed based on the yolk, we have different types of eggs.
Yes, ma'am.
Yeah. So here you can see in the sea urchin we have a little amount of yolk, whereas in the frog we see some more amount of yolk, which is mainly concentrated in the vegetal pole than in the animal pole. This is the chicken egg, where central vesicle we have discussed, where it is in the center of the egg. So, egg with moderate to little yolk undergoes holoblastic cleavage. That means sea urchin or frog, where the yolk is medium or low. Okay, so it is holoblastic means it is the complete cleavage. Okay, And in sea urchins, a symmetrical blastula is produced surrounding spherical blastocoel. In amphibians, we will see asymmetrical blastula, okay, and the blastocoel will be displaced to some other portion because it is having more yolk, okay. So this is how it looks. So here we have the below one is the vegetal fold and the top one is animal fold. And we have blastocoel, which is like a cavity where the fluid is filled, okay, fluid filled cavity. And the cleavage patterns are where you see, as we have discussed, like in eggs where the little amount of moderate amount of yolk is present, that will undergo complete cleavage, whereas the eggs with large amount of yolk undergo meroblastic cleavage or incomplete cleavage. Okay, this we can see in reptiles and birds. And clear cytoplasm is seen in one fold, and that is called as a blastodisc. Okay, so this
Okay,
is how
ma'am.
it looks like. Yeah. So at the center we have the yolk, and the embryonic cells will start cleaving at the top of the yolk. Now finally, it will form a blastocyst stage. Okay, in mammals, this blastocyst has an outer single layer of cells which are called trophoblasts. They usually form the placenta. So in human beings, we can see the placenta, right? So which grows along with the baby. It's a temporary organ which is formed only during pregnancy. And we have blastocoel, which is the fluid-filled cavity, and the cells which are present inside, they are called inner cell mass, which will form into the embryo. So now here, this pink color one is in mammals, we have inner cell mass blue color cells, and the outer layer is trophoblast, and the center cavity is blastocoel. And the other one is in birds usually, where the yolk is present in a large quantity, yellow one, and the blastodisc is present at the top. Now, what is the fate of the blastomeres? So here the fate means, now at the embryonic stage, every cell has the potential to become every other organ. Okay, there is nothing like this cell has to become this organ or like that. But once at certain stage, their fate will change. So they have to decide. They have to decide means here there is no intelligence here, but there are some signals which will decide which cell has to become what. So in mammals, early blastomeres, they don't have any committed state. So every cell has the potential to become all a complete organism actually. But whenever they form the eighth cell stage, the outer surface of blastomeres, they will become a flat by a process called compaction. And now they will divide asymmetrically. Okay, the division is not symmetrical or uniform. Once this happens, then the next stage is, now you have a ball of cells. The next stage is rearrangement of these cells. Now these cells will rearrange themselves to form three germ layers by a process called gastrulation. And these three germ layers are ectoderm, mesodome, and endoderm. Ectoderm cells are present in the exterior of the cell. Then mesodome is at the center, and endoderm is the innermost layer. So from the ectoderm, we have epidermis of skin, nervous system, and sense organs which will be developed from the ectoderm. Then in the mesodome forms skeleton, muscles, blood vessels, heart, blood, gonads, kidneys, dermis of skin. So all these are formed from the mesodome. Then endoderm, which will form lining of digestive and respiratory tract. Then liver, pancreas, thymus, thyroid, so all these things. So during the process of gastrulation, a variety of changes will take place in the cell. So one is invagination, involution, delamination, and ingression. invagination is nothing but the cell sheet will bend inward, and involution means they will roll inward, and delamination means they will split the embryo into two, delaminating, and ingression means the cells break away from the cell sheet and they will migrate as the individual cells. From there, the rearrangement actually takes place. So this gastrulation process is also differs from organism to organism. So here it depends on the amount of yolk. So in sea urchin, first what happens, it begins with the formation of vegetal plate and ingression of primary mesenchymal cells into blastocele. So from the blastocele only it will start. From blastocele it will bend inside and it will form a tube-like structure. And the remaining cells of the vegetal plate invaginate into blastocele, forming the endoderm. Here we see archenteron. Archenteron, yesterday we have discussed, like in frogs usually, archenteron forms the future gut, okay, digestive gut. So the cells which are present on the surface of this archenteron, they will form ectoderm, and the outer cells are they will form as endoderm, okay, in the blastocele. So here you can see how it looks like. Like the top ones are original ones, and the below ones are like graphical representation for your understanding. So here the yellow cells are future endoderm, and the top one blue cells are future ectoderm, and this is vegetal pole, and the top one is animal pole. Now from the blastomere, the cells will bend inward, and they will form it that like a tube-like structure where the bottom of this tube forms anus, future anus. Okay, this is how the shape will be formed. Then in frogs, okay, cells from the animal pole involute over the dorsal lip of blastomere into the blastocele, and finally the cells will press against the wall and they form archenteron, and these movements create two layers, outer ectoderm and inner endoderm. Mesoderm will be formed later in between this ectoderm and endoderm. So here you can see we have animal pole at the top, vegetal pole at the bottom. We have the dorsal lip of the blastomere, which is pink in color, just below that. And we have blastocele, mesoderm, ectoderm. Now, this foldings of these layers will finally form archenteron, yolk plague, all these things. And this invagination will form neural plate and neural fold. So it is not a single event. Continuously the foldings, rearrangements, they will be taking place. So finally they will form the structure of the embryo. Okay. Then in birds, they form disc of cells which are called blastoderm, and they first form two layers. Okay. And in between these two layers, blastocele cavity is formed, and finally three germ layers are formed, which are primitive. First, a primitive streak is formed. From primitive streak, all these three germ layers, ectoderm, endoderm, and mesoderm, will be formed. So here you can see the blue one is blastoderm, and below that is orange one, and below that yolk. And between this blue and orange layers, we have the blastocele, which is in between. And that structure is called as primitive streak, the pink structure which is formed in between. And that can form into mesoderm. So we have ectoderm, endoderm, and mesoderm. And in mammals, it is similar to the birds. Okay? And the inner cell mass will flatten here, and it will form two layers. Okay? Then finally, the third germ layer also will form. So the outermost layer forms trophoblast, and we have inner cell mass. Blue color one is inner cell mass, and the outer layer is trophoblast. And in the inner cell mass, it will form an amniotic cavity, so which will hold the amniotic fluid. and it will form yolk sac in between. And that blue color amniotic cavity will form the ectoderm, and below that it will form endoderm. And at the center, we will see primitive stick where the mesoderm is formed. Then, based on the adaptation, in some animals they will live in some extreme conditions, maybe dry regions, okay, like that. So to adapt, they will form some extra embryonic membranes. So what are these extra embryonic membranes? They are amnion, chorion, yolk sac, and allantois. So this also we have already discussed. These fetal membranes. So amnion encloses amniotic fluid. Chorion is located near the eggshell of the bird. In mammals, they will form the placenta. In the yolk sac provides food in the birds, whereas which is not necessary in mammals because placenta is present. And allantois is mainly for gas exchange and for the elimination of waste. Okay, and in mammals, it will contribute for the blood vessels in the developing umbilical cord. So this is how it looks like. It is a chick embryo where the outermost is the chorion and below that is amnion and the other side it is yolk sac and below it is allantois. Whereas we see chorion, amnion and little yolk sac is present. But here we see chorion villus and umbilical cord blood vessels. So because we have placenta, so yolk is not much needed. Okay. Now finally the organogenesis. Once we got ectoderm, endoderm and mesoderm, from that we will get different types of specialized cells and finally organogenesis takes place. So organogenesis is the formation of organs in their proper location. It is mainly through the interaction of the cells within and between the three germ layers. Okay. So after gastrulation, this organogenesis takes place. So the cell's location in the developing embryo will determine its fate. Okay. So where it is located. So based on that, its fate will be determined. That is called cell determination. And this cell fate is established in two ways. One is by the cytoplasmic determinants or the chemicals which are present in the cytoplasm. Or second is the interaction with the neighboring cells. That is called as cell induction. So other cells which are located nearby also contribute. So we'll see some examples in the Drosophila where which is not much necessary to remember, but it is just for your understanding. So we have already told about homeotic genes, the master genes which will regulate this development. So in this, the best example is the salivary gland development, where we have SCR gene, sex hormones reduced gene, which is a homeotic gene present in the antenna pedia complex. So before the organogenesis, it is expressed in anterior band of the cell. But at the same time, DPP, another gene, which is released from the dorsal cells. So anterior cells are releasing SCR gene, dorsal cells are releasing DPP gene. So they will form a gradient in the dorsoventral direction. Gradient means unequal distribution. At one point it is very high concentration, and another point it is low concentration. Now, DPP inhibits the formation of salivary gland rudiments. Thus, during organogenesis, salivary gland develops in the area where SCR is expressed and DPP is absent. Okay, so this is a very good logic and very like beautiful explanation. Like as DPP is stopping the formation of salivary gland, only where the DPP is not expressed and SCR gene is expressed, at that portion the salivary gland is formed. So this is a signaling, okay, where the cells have to differentiate into what type of organ. So here you can see prior to organogenesis, we see some band-like structure, okay, where the DPP. So you see on the top it is thick blue, whereas at the bottom it is light blue. That means it's a gradient. Where it is thick blue, the DPP concentration is very high. Where it is light blue, the DPP concentration is reduced. And where it is completely reduced, there the salivary gland will form. Okay. So this is in the Drosophila. Whereas in vertebrates, it will form notochord and dorsal nerve cord. This process is called neurulation. And this notochord, it forms from mesoderm. And the region of dorsal ectodermal cells situated above the notochord, they form neural plate. And the cells which are present in the neural plate, they form a long cylinder which is called neural tube, which will finally become brain and spinal cord in the organism. So this is how it looks. So the neural plate is at the top, which forms from the ectoderm, and below that, that blue color, then below that we have pink color which is from mesoderm, it has notochord, and below that the yellow one is endoderm, and below that is yolk sac. This neural plate will fold to form neural groove and neural folds. And this in the mesoderm, this notochord will form a spherical structure, and at the center you have the notochord. And below that, then once it invaginates the neural groove, it finally forms a neural tube, and to that on both the sides of the neural tube we have neural crest, and this on the two sides of the notochord they form somites. So what are the functions we will see? So the generation of somites. So the mesoderm sheets on either side of the notochord they separate into a rounded region called somatomeres. They are separated into segmented blocks called somites. They form anterior-posterior wave, okay, and they will give rise to skeleton, muscles, and connective tissue in the near future. And the mesoderm in the head region, which connects to the somatomeres. So those where the mesoderm is connected to somatomeres, they will form into face, jaw, and throat. And some other body organs also develop in the mesoderm and somites. Okay. And now this mesoderm is separates into two layers, and the coelom is formed in between. So here you can see. Now the central mesoderm, which is called chorda mesoderm. This chorda mesoderm will form notochord, and the intermediate mesoderm, they will form kidney and gonads, and paraxial mesoderm will form head and somites. And this lateral plate mesoderm, they will form circulatory system, lining of body cavities, and extra embryonic structures. And the somites, they further form cartilage, skeletal muscle, and dermis. Then the neural crusticles, which are present on the two sides of the notochord, they will they are nothing but the neural groove closest to the form of neural tube, and there are small cluster of cells called neural crusticles. They migrate to many different regions of the developing embryo, and they will usually prefer three pathways. First one is cranial neural crust cells. That pathway, if they migrate in that pathway, they will form head and neck. And second is trunk neural crust cells, which are present in the posterior region. They migrate into ventral pathway cells. Then they will form sensory neurons and Schwann cells. If they enter into lateral pathway cells, they will form melanocytes of the skin. So this is how it looks. The anterior region and posterior region. And those which are present on the anterior region, okay, anterior somites, and those which are present in the posterior region, they are posterior somites. So these posterior somites usually enter into two pathways, lateral pathway cells that forms dorsolateral route between the epidermis and somites. And the ventral pathway cells, they travel ventrally through the anterior half of each somite. Okay. So no need to remember in depth, just remember from which cells, which pathway gives what type of cell. Then if there is any mutation in the neural crust cells, then that particular the gene which is expressed or the organ which has to be formed, that will be not properly formed. So I told you they will form the melanocytes. So therefore, if there is any mutation, the melanocytes of the skin, they don't form, and you can see the white patches in the baby as well as in the mice. So many of the unique vertebrate adaptation involve this neural crust derivatives. Examples are, that means based on their adaptation to the environment, they will change these neural crust cells. The first one is gill chambers. They are in fishes, you can see, they are very important for gas exchange. So, and also they will give the high metabolic rate in some animals which are required. And other is for better prey detection. They will make some changes in the sensory perception also. Okay, so these are some modification in the neural clusters. And this is the final, like, which cell feels what. Okay, so first the zygote forms blastula, ball of cells, then they will undergo three layers, which is called gastrulation. So after gastrulation, we get ectoderm, endodrm, and mesoderm. So first in the ectoderm, it will form epidermis, skin, hair, epithelium, inner ear, and lens of eye, which will further develop into dorsal nerve cord in cortex and brain, spinal cord, spinal nerves, neural crust cells in vertebrates, and neural crust cells further develop into gill arches, sensory ganglia, Schwann cells, adrenal medulla in vertebrates. Then forms mesoderm. In the mesoderm, it forms integuments, gonads, kidneys, circulatory system, somites. The integument will form the outer covering of the internal organs and lining of thoracic and abdominal cavities. And circulatory system form blood vessels, okay, blood and vessels. And in caudate, this mesoderm forms notochord, and the somites form dermis, and also in caudate it will form striated muscles. Whereas in vertebrates, they form heart and skeleton. Then endoderm, which forms pharynx and major glands like pancreas, liver, the inner lining of respiratory tract and digestive tract. So this is how the after gastrulation and organogenesis takes place. The next is vertebrate axis formation. So as we have discussed, there are two scientists. One scientist we have already discussed. So Hans Sprayman and Hilde Mongold. Okay. They transplanted the cells from the dorsal lip of one embryo into the belly region of the another embryo. And we already discussed, like, the two notochords are formed in the second embryo. And here you can see like this. So the dorsal lip is transplanted to here. And you have a primary fold and secondary neural fold. And finally, from the dorsal, from the belly region, another embryo, another neural fold or neural tube is formed from where the another embryo is formed. So therefore, this is the primary organizer of the organogenesis. So an organizer is a cluster of cells that release diffusible signal molecules which will convey the positional information of the other cells. So that's why I told, like, they will secrete some chemicals which will determine the position of the other organs. They are called as morphogens, whose concentration will give us some information like where the organ should be present. So this is how they have made a graph. This is a morphogen, the top one, where we see blue, green, and brown color. So the blue color, if it is high in concentration, it will result in the organ A. Okay. If green, it is bit lower than the blue one. So then it will give the organ B. And C is still less. So it will give the organ C. So like that. Based on the concentration of the morphogen, we get different organs and their positions. So creation of the stamen organizer. So in frogs and fruit flies, during oogenesis of the mother only, the stamen organizer will be formed. So this maternally encoded determinants or chemicals which are localized in the vegetal pole of the unfertilized egg will determine, they are nothing but they are the organizers in determining the structure of the organism. So this is how it looks. In the vegetal pole, we have the green ones, which are dorsal determinants, which will migrate and they will determine the structure or overall morphology. And in some organisms, we have Wundt signaling pathways. So there are various signaling pathways also which are involved. So Wundt signaling pathway is one which is seen in vertebrates and invertebrates. It will affect the morphogenesis or organogenesis. So what is the function of the Spearman organizer? So here dorsal lip cells do not directly activate dorsal development, but dorsal mesoderm development is a result of the inhibition of ventral development. They are not like signaling the dorsal development, but they are inhibiting the ventral development so that the development shifts to the dorsal region. And the bone morphogenic protein 4 is expressed in the marginal zone cells of the frog embryo, and this acts as a morphogen, and it is a it is like a morphogen which is secreted by this Spearman organizer. Okay. So this is how it looks in the animal pole and vegetal pole. We have the mesoderm at the center, then epidermal ectoderm, and in between neural ectoderm, and below yellow one is endoderm. So all these evidences indicate that the organisms are present in all the vertebrates, and in chicks we see from the primitive stage we will get a Henson's node which functions like a Spearman organizer. It produces some molecules and they are involved in the development of the organism. Okay, same thing you can see in the frog also. So it is nothing but, see, every organism it is different. Okay, we cannot detail study each and every organism. But the thing is, there are always organisms, primary organisms from there which secrete some molecules, and the concentration of molecules will determine which cell has to become what. Once the cells get differentiated, then they will form into some tissues, then organ systems, and finally organs. The perfect axis has to come. And the induction, like how the cells influence the other cells. So this is again of two types, primary induction and secondary induction. So primary induction occurs when the three germ layers are formed. Whereas the secondary induction is once the layers are formed and the tissues are developed, in between the tissues, so that coordination is by secondary induction. Okay, this is mainly in the development of lens, eye lens in the vertebrate. So we will see the example. So this is the outer one is the ectoderm, and inside it is neural cavity, and to that we have wall of forebrain. So this optical stalk which is present, from there this ectoderm will invaginate and it will form a lens vesicle, and whatever the wall of forebrain is there, that will invade inside to form optic cup, and this lens, this optic cup, from that optic nerve is developed, and then lens, sensory layer, pigment layer, all are formed, and finally it will form the retina. So it is nothing but the lens cells are influencing the forebrain cells to finally form the retina. Okay, so this is about the induction and since fertilization to development of organisms. So what are the events are there that we have discussed? Okay, if you want important questions, I will just post it in the group. Okay, because right now I don't have the syllabus. I will look into it and I will post it in the group. If you have any doubts, you can ask.
Okay, ma'am.
Okay, is it clear?
Yes, ma'am.
Yes, ma'am.
Okay, then I will post the questions in the group. Okay, thank you.
Thank you, ma'am.
थैंक यू मैम। अरे चल गए जल्दी मैम। चल जा रहे हैं, मेरे पजामा भैया।